cytokines are very important. For example, the stromal cells of the bone marrow, they produce IL-7, which contributes with the survival of T lymphocytes that are produced there. On the other hand, for the B cells, we have the BAFF factor, or BAFF, which is a cytokine from the tumor necrosis factor family, TNF-alpha family proteins. So they contribute with the survival of B cells as well. So we have these cytokines that they bind in their respective receptors and activate these T cells or the B cells to remain alive and metabolic active rates. So despite the fact that the naive lymphocytes might survive between 1 to 3 months, the total numbers of our repertoire of lymphocytes is fairly constant. And the reason why is that it's because our immune system is able to detect the drops in the numbers of lymphocytes and to induce proliferation again, especially in the stem cells residing inside of the bone marrow. So this mechanism to reconstitute the number of lymphocytes in our body to remain constant is called homeostatic proliferation. So what are the effector lymphocytes? They are properly the ones that are going to fight an infection. So once these naive lymphocytes, they are introduced to the pathogen or the fragment of a cancer cell in the lymphoid organs, they become effector cells. And effector cells, so T lymphocytes, they might be effector CD4 cells or CD8 cells. CD8 cells, once they are active, they become cytolytic. So T lymphocytes that express CD8 marker in the surface, they can kill other cells. They will become active. They will start now expressing molecules in the surface of their cell membrane that will allow them to migrate to the inflammatory tissues where the inflammation is happening, when the inflammatory process is occurring, and they will infiltrate there. So they acquire an active phenotype to destroy cells. That's why they are called CTL, cytolytic T cells, okay? And CD4 cells that are supporting the inflammation, they are T helper cells. They can be T helper type 1 if it's an intracellular infection. They can be T helper type 2 if it's an extracellular infection. If the infection is related to fungi or a specific cancer inflammation process, a TH or T helper 17 type, and so forth. So once the naive lymphocytes are activated, they enlarge in terms of volume and they proliferate. They start producing a lot of proteins and specific factors inside that are responsible to contribute with the inflammatory process. They grow in size and volume as well. So as we can observe here in this figure, like the morphology of the lymphocytes once they are activated, they start producing and secreting antibodies, not before. When they are naive, the antibodies are just expressed in the surface of these cells, right? But when they are introduced and activated to a particular antigen, to fight a particular antigen, they drop in the circulation and they are transformed into plasmoblasts or plasma cells. So now they are secreting a lot of antibodies. This constitutes a humoral response. It's an immune response constituted by soluble antibodies that are dropped in our bloodstream to neutralize a particular pathogen. They are coming from activated B cells. They are activated in the lymphoid organs and they acquire a new gene expression program that will tell them, now you need to secrete the antibodies. You not only keep the antibodies in the surface, now you secrete them. And then they will secrete different types of antibodies depending on where they are, if they are in the blood, if they are in the mucosas, and so forth. In our bone marrow, there are other cells that are produced from the progenitor, from the myeloid lineage. They will not become lymphocytes. Some of them, they have like similar characteristics compared with the lymphocytes, but without the T cell receptor, okay? But they will still have an active behavior similar, for example, to a T lymphocyte. So one example is the natural killer cells. Natural killer cells, they are very similar to the TCD8 cells, but they don't have the T cell receptor. They have similar morphology and they will also attack infected cells or even cancer cells. by secreting their poisons like granzyme B, perforin, so these enzymes that can destroy the cell membrane of an infected cell or of a cancer cell. There is also another type of myeloid cell that is similar to the lymphocytes. It's called cytokine secreting innate lymphoid cells, or ILCs. So these cells are very interesting because, as opposed to the natural killer that is similar to a CD8 type of T cells, these ILCs, they are similar to the TCD4 cells but lacking the T cell receptor. So there are essentially three different subsets of ILCs that we can discuss later, but they are differentiated, or they are grouped in terms of the cytokines that they produce, in terms of the chemicals that they release to modulate the immune response, similarly to what happens with the CD4 T helper cells. ILCs are mostly found in all tissues in our bodies, and especially in the mucosas, but they are rarely found in the blood. They are very similar in terms of functions to the CD4 cells, but they lack these receptors. So they are considered to be cells from the innate immunity, okay? Thank you very much. If you have any questions, please don't forget to subscribe to the channel and leave your questions in the comments below so I can read it and reply to you. And see you in the next lecture. Bye-bye.